Okay, welcome to the seminar today. We are pleased to have Dr. Benjamin Carone today, fresh back from sabbatical. Dr. Carone spent his undergrad and PhD work at UConn, right? And then moved and did his postdoc in transgenerational inheritance and epigenetics, a theme that we'll see followed, at University of Massachusetts Medical School, and then did a visiting assistant professorship at Williams College. So he spent most of his time up in New England, and then he's down here in South Jersey, but this past year he moved a little bit further away and spent a good amount of the year in Australia. And we're all looking forward to bringing the Tasmanian tiger back.
It's back.
Oh, it's back. It's here on campus. As Dr. Carone. So please welcome me in welcoming Dr. Carone back to Rowan and here for today's session.
I can honestly say it is really good to be back. I did miss everybody this past year. So I wanted to start off today's talk, first off with a thank you. Getting the opportunity to do a sabbatical is a really generous thing by the university. It's really rewarding professionally and personally. And I hope you'll see that I took the most advantage of last year. I just wanted to thank the biology department for supporting it, the dean's office and the provost's office for supporting this for the past year. My two kids who had to wear school uniforms in Australia, that was a new thing for them. My students in the laboratory, including Juliana, Yesenia, and Lauren, who are just absolutely phenomenal while I was away. It was sort of a surprise that I was going to be gone for a year. Good luck. They did wonderful. Dr. Andrew Pask and Dr. Rachel O'Neill. Dr. Andrew Pask is at the University of Melbourne, and Dr. Rachel O'Neill is at the UConn, who's the sequencing core. And then, of course, Chris Hemsworth, who you may or may not know actually financially supports this project. He reached out to me several times when I was in Australia, but I was too busy doing research to meet with him, but hopefully in the future I get an opportunity. And last but not least, your word of the day is antipode. So antipode means the exact opposite side of the planet. So it's not this way, but this way. And that's pretty much where we were. So I spent most of the last year at the University of Melbourne, which is in the southern part of Australia. The climate's very similar to Philly, but is the opposite side of the planet. Okay, so that was pretty cheery. We're going to go a little bit dark now. In the past 600 million years or so, there have been 5 major mass extinctions. Mass extinctions are defined by greater than 75% loss of species over a short geological period of time. The first two were about 350 and 450 million years ago, the Ardovian and Devovian events. These are both precipitated by climate change, rapid heating and cooling of the planet. Lost more than 75% of these species in a couple million years. The next major events were the Permian and Triassic events. These are precipitated also by climate change, but ultimately driven by volcanic activity, large amounts of CO2 being released from the ground. We're all familiar with the asteroid that hit the Yucatan Peninsula about 65 million years ago that killed most of the dinosaurs and most of the species on the planet. This species loss was again greater than 75%, about 2 million years. And these are the 5 major mass extinctions. Sorry. But many scientists now consider we're in a 6th mass extinction called the Anthropocene. And as you can expect, mostly precipitated by human beings, potentially Justin Bieber. We consider this, it's not well defined. However, if you look at recent rates of extinction, and this is the rate of extinction over a million species years, the amount of length that we're actually investigating here, and this study is for Barnowski et al. In Nature. Over long periods of time, we end up around 1.8 or so extinctions per million species years. If you look at the more shorter time spans in recent years, you can see an extremely rapid rate in the increase in the number of extinction events. And if you look at the vulnerable, endangered, and critically endangered species, it's even higher. So all of this is to suggest that these higher rates, we are in a period of time in which we're losing a lot of species, right? So potentially a 6th mass extinction. You kind of look at the numbers of these things. It's estimated that about 30,000 species a year on average are being lost, about 150 a day. So a tremendous amount of diversity. And the World Animal Foundation predicts that about 50% or more of these species will be lost by 2050. So it's a major problem, obviously. This was not precipitated by volcanic events or climate change that's naturally derived. It seems to be the most recent thing that's changed dramatically has been that humans have come onto the scene. So since we've been around the planet doing our thing, the world's lost about 50% of its original forests. More than 40% of Earth's land is used by human beings for commercial, agricultural, or residential usages. CO2 concentrations are the highest they've been in 650,000 years. We've introduced invasive species in pretty much everywhere we wound up. And if that wasn't enough, we've pretty much directly killed everything we've come across in the past couple hundred years. So we're not doing too great for the planet. We're doing especially poor for Australia. So this talks about marsupials in Australia, but it's kind of interesting because Australia is a especially sensitive place on the planet. Out of 273 endemic land mammal species, more than 10% of them are extinct, more than 20% of them are endangered, and even more are threatened. So this is not just true for mammals, but it's also true for birds, reptiles, frogs, and everything. But Australia represents a very sensitive ecosystem, which has taken tremendous impacts from human presence. So what can we do about it? There are some solutions. I will say that if you get to know me, I'm actually an optimist. I feel pretty good about human beings are trying really hard now. Look back at the 50s and 60s, Industrial Revolution, things were really, really bad. So we're trying to clean our act up. We're not doing great, but we're doing something. By reducing pollution, increasing sustainability practices, reforesting where we can, and protecting specific fragile ecosystems, but designated national parks, things like this. So in terms of when we actually target a species, we do really well. So when things go on the endangered list, they can come off the endangered list if we intervene. And so, for example, the gray wolf was near endangered or was endangered. It was reintroduced to Yellowstone National Park, and now it's part of a thriving and healthy ecosystem. Introducing that predator to that ecosystem actually helped and benefited that environment. All these other animals, including the bald eagle, mountain gorillas, black rhinos, and humpback whales, are now also off the endangered species list. So they're vulnerable, but their populations are rebounding now that we've decided not to kill and eat and remove them from the environment. So that's great, but what about species that have already gone extinct? Is there a solution? And what I'm going to tell you a little about today is hopefully we can use modern genetic engineering technologies to try to de-extinct or at least bring back some of the core features of these to reintroduce into the ecosystems. So this wasn't my idea, obviously. This work has been in the scientific community. Ideas been in the scientific community for a while. It was most recently commercialized and funded by a group called Colossal Biosciences. It's actually an American-based company in Texas. It was founded by Ben Lamb, who is a serial entrepreneur who started a number of tech companies, and then I think he decided he really liked Jurassic Park and being a good environmental steward and created this company. And I think it's important for everybody to consider here that when we're talking about the idea of de-extinction, we're not actually talking about bringing the Tasmanian tiger back exactly as it was. But we're more about talking about bringing back something that has those core traits of that animal that fit into that ecosystem at the time, so it can help balance our current ecosystem. Ideally, that's exactly a Tasmanian tiger, but that's not entirely possible. The reason why that's not entirely possible, a good example would be something like, imagine that we all go extinct, not a good scenario, and in 10,000 years, they find my DNA. They try to take that and recreate human beings based on it. Well, they're going to look back and say, okay, well, there's a moderately attractive, somewhat stout guy who likes to walk on the beach, who's white and has these features. But that doesn't represent all of us, for sure. So you're not going to be able to capture the genetic diversity of a population later in time. So we have representative examples of the thylacine, but again, I don't want everyone to think that there's going to be the tigers roaming around exactly as they were. The idea is to sort of help be good environmental stewards by bringing back these core traits. Okay. Maybe not. Yes. Okay. Okay. So the key three animals that Colossal Biosciences is focusing on are the woolly mammoth, the dodo bird, and the Tasmanian tiger. Common to all three of these is they've all gone recently extinct. They've all been extinct by the hunting for the hunting of humans. And we have DNA sequences for all these organisms. So we know the sequence of them, so we can potentially reconstruct that code. The woolly mammoth went extinct about 10,000 years ago. It was hunted by humans as it came to smaller and smaller regions as the climate was changing. The dodo bird had a very short tenure. It lives on a small island in the Indian Ocean called Emeritus. And we discovered this island as explorers around 1600. And by 1660, we had eaten all the dodo birds in that island. And then, of course, there's the Tasmanian tiger. This guy was alive up until 1936 in a zoo. It's endemic to Australia mainland and Tasmania. This is a colorized video, so it's pretty neat, from the National Film and Sound Archives. So they took a black and white video and they tried to add some color back to it so people could kind of appreciate it. It's a marsupial, obviously. It has stripes on the back. It's neither a cat nor a dog. It's a marsupial, even though it's called a tiger. Some people call it a Tasmanian wolf. It has this crazy jaw. All these carnivorous marsupials have the same thing. And it's really important to the Australian people. So if you ask Australians, I think this is a good idea. Everybody's on board. Everyone's really excited about this. It sort of represents something key about the continent. So the group that I was working with in Australia is obviously focused on the Tasmanian tiger. There are other groups working on the other two animals as well. The group I was working with includes or is housed in what's called the Tiger Lab, or the Thylacine Integrated Genome Restoration Research Lab. It's led by Dr. Andrew Pask here. He is a professor of epigenetics at University of Melbourne. He was working on the thylacine for long before Colossal Bioscience became interested in it. He was actually approached by Colossal Bioscience and says, can you do this? And as an epigenetic scientist, a developmental biologist, and someone who's working on this animal, he said he gave it the old Australian, let's give it a go. That's a classic. He now leads this laboratory. It has 60 people. It's funded with $40 million. It's roughly the size of our building in terms of the scientific capacity. And this is where I spent sort of most of the last year. I should say that I got kind of lucky. I've known Andrew for 20 plus years as a friend now. And I called him up and said, let's do a sabbatical. And he said, boy, do I have something for you. So it's been a really great experience. All right, so luckily, he published instructions on the website. So if you want to look at how a Tasmanian tiger is brought back to life or de-extincted, you can go to the Tiger Lab website and follow this nice flowchart. The key features that are required for this kind of thing are, first, you need DNA. And so this is why we're only doing, or you can only do really the things you have DNA for, like the mammoth, the dodo, and the Tasmanian tiger. There are museum specimens of the Tasmanian tiger that are still in existence with remarkably well-preserved DNA. We're going to identify the differences between the Tasmanian tiger genome and a closely related marsupial species, the fat-tailed dunnart, sequence this genome, and identify the differences. Once we know these differences, we can then take cells. The type of cells I'm going to discuss in detail as different kinds of cells are applicable for different reasons in this project. But take the cells, put the Tasmanian tiger DNA inside of it, fuse it with an empty dunnart egg, create an embryo, put it into the Tasmanian devil, I don't know why they decided that one. You'll see in a little bit. Isolate the animals, and then you'll have a tiger. Now, it's a fairly simple flow chart, but all these things are incredibly complicated. They're expecting around 10 years to get through this, to finally create an animal that has a lot of the traits of the Tasmanian tiger. Key things that have been accomplished so far, and both of these are from the past laboratory. The Tasmanian tiger has been sequenced. We have a complete genome assembly. It's all set. The fat-tailed dunnart has been completely sequenced. There's a complete genome already assembled. So steps 1 and step 2 have been done. Step 3, which is to compare the genomes, is something that I'm working on with the group as well. And today I'm going to talk about strategies for step 4 and step 5 that I was actively working on in Australia. So the proverbial elephant of the room here is, why would you take something that's about the size of a dog, right, 50 pounds or so, top apex predator, and somehow take a small mouse, like this is about the size of a mouse, the fat-tailed dunner, and convert this, the genetic engineering, into this. This is probably the biggest conceptual hurdle that most people have in this project, and of course it is fairly challenging. But in order to understand why this model was chosen as sort of the host or surrogate for this genome engineering project, you have to know a little bit more about what these animals are. So, key to this, both of these are marsupials. So as you guys have studied over time in the biology program, there's a whole bunch of different flavors of animals. Some of these animals are mammals. In this group of mammals, we have monotremes, which are egg-laying mammals, marsupials, which are mammals that got pouches, and the rest of us eutherians, mouse and human beings. This tree represents how similar we are to each other. All marsupials are more similar to each other than we are to a mouse or human. So while it might seem like a good idea to do this experiment in a dog, for example, the genome of a dog and the genome of the Tasmanian tiger are very, very different, despite them looking very similar. But in actuality, the genome of this small little mouse kind of thing, fat-tailed dunner, is much more similar to the Tasmanian tiger than it is to the mouse or anything that might look like it. Even more so, if you look at the clade in which all these things are in, the dunner and the Tasmanian tiger are actually only separated by about 40 million years of evolution. This is actually pretty small. So both these genomes are about 90% similar or more. Despite the size, they share a lot of common traits, especially in terms of the developmental profiles and how they develop. And then finally, one of the things that's really key for this entire clade is that they all have 7 chromosomes, and that's actually really rare. You start looking at different chromosome numbers in different organisms. Humans have 46, mice have 40. You can't mix and match those things together. The different number of chromosomes really screw things up. So in this case, having the same number of chromosomes, similar genome architecture, the actual genetic engineering is actually much more feasible in this case. Still a stretch. So if you were to do this, genetic engineering, the way you would do this is through cloning. Cloning has been around since the mid to late 90s. The first example of a cloned organism was Dolly the sheep, and this occurred successfully in 1996. And this was done by something called somatic cell nuclear transfer. And I'm going to outline this process a little bit because this is actually how we're going to try to do this with the thylacine. And in this process, we take an egg cell from a donor. In this case, this was all from sheep, so the same organism. In the case of the donor and the thylacine, these would be from different organisms, which just has not been done yet. So in this particular example, we would take an egg cell from a sheep. We would pull out the nucleus of the DNA. It's called an enucleated egg. We would take a differentiated somatic cell, so like an adult cell, and Dolly, that was a mammary gland. Take out the DNA of the nucleus. We can fuse these two things together. In this case, it was injected. In some cases, it can be fused. This egg will reprogram the DNA inside this nucleus to create what's called a totipotent embryo, so an embryo that can become anything, implanted into a female and grow an adult animal. This has also been done with primates, but not been done with humans. So even though it looks pretty simple on a cartoon, it's actually very challenging. It requires eggs from a living host, so we will have this with the donor. It requires intact DNA from donor cells. We do not have this for the Tasmanian tiger, nor will we ever. So that's an important caveat to this. We have a sequenced genome, but we don't have intact nuclei to do this. Requires a recipient animal. We do have a similar marsupial, the dunner or the numbat. There's a bunch of other ones we could use. And then finally, the last challenge, this is kind of the work that I'm focused on as well, is that incomplete reprogramming of epigenetic states for this nucleus can actually be a major hurdle in this process. So if you want this to actually work efficiently, we need to learn about the epigenome and how to reprogram it. And so what does this look like? So one of the most shocking facts that I usually tell in my classes is that almost every single cell in your body has the exact same DNA, despite being very different. I think that's very confusing to a lot of people because you think, well, cells are so different. Liver cells are completely different from muscle cells. How do they have the exact same DNA? All right, and the reason they have the exact same DNA but can be different phenotypes is because they actually express different genes. So early on, when you're first made, this is the earliest stage of an embryo, so zygotes, then about 16 to 32 cells in the morula. All of these cells are exactly identical. They have the same epigenetic state, and they can become anything. So they're stem cells. These inside the blastocyst, this inner cell mass, are also totipotent. They're also stem cells. These type of cells can become any of these different cell types just by expressing different genes. So liver gene is turned on for these cells. Muscle genes are turned on for these cells. This process is largely facilitated by changes in gene expression, but also changes in epigenetic states. So there's a lot of different epigenetic marks today. I'm just going to talk about one of them. This is the one where we've started to investigate, which is histone modifications. And even though you have the same DNA inside your cells, the DNA itself is actually wrapped around an octamer of histones. These are actually just proteins. And these proteins can actually be chemically modified. And so these tails on these histones can be marked by different chemical modifications, including methylation, acetylation, ubiquitination. And those markings can actually instruct the genome whether or not these pieces of DNA should be turned on or turned off. And so, of course of development, as you go from early stages of morula all the way down to these differentiated cells, you acquire a series of different histone modifications, which effectively lead to an epigenetic program that is established. Okay, so we're going to get into, I have two projects that I'm going to talk about today that I worked on in Australia. The first one is one that was really already existing that I jumped into, but I'm still collaborating with. It's really exciting. And the second one is one which sort of I initiated, and both of them are ongoing here, but this first one, a lot of this work had already started for a long time before I began. This is supervised by a research scientist, Jed Taruli, a graduate student, Pranav, who is also my PhD student that I co-supervise. And I also want to thank Ebony, who I worked with a lot over there, and Beth is the lab manager. They have 60 people, so it's an incredible group. But these individuals were directly involved in generating a lot of material for what I'm about to talk about. So the first thing we were going to do, as opposed to enucleating an egg, for example, and having an intact somatic cell from the thylacine, we don't have that, is to take some thylacine DNA and put it into stem cells that we could actually use to put into an egg. So I filled in this term here with stem because the first thing I'm going to talk about is how to modify a stem cell that we can put the thylacine DNA into and create our thylacine embryos for this process. And so to create these stem cells, we're going to use a revolutionary technique called induced pluripotent stem cells, or iPSCs. For those of you who are alive in the year 2001, which is dwindling in this room, George W. Bush was the president at the time, and he actually banned the creation of new embryonic stem cells. Okay, so this time he said stem cell research is not a viable thing that we're going to pursue. And so it led people really to think about, well, can we make stem cells instead of actually trying to derive them from embryos? And so this group led by Shinya Yamanaka, and his student Takahashi actually published this paper cited over 33,000 times, and they were able to take differentiated cells, mouse embryonic fibroblasts. They added 4 key reprogramming factors. So these are transcription factors that are present only in stem cells. They were able to add these to these cells and then convert these differentiated cells into what's called IPS cells, so induced pluripotent stem cells. And that's these kind of like colonies, we call them here, or embryoid bodies. And these look very, very similar to what embryonic stem cells look like over here. So you're going from this to this. The reason why this is important and what makes these stem cells is if you then take these cells or these ones that you've made in the laboratory and you inject them into an early developing embryo, in this case tagged with GFP, they can actually become any type of cell in a developing embryo. And so you can imagine very easily, if you can do create this cell line, do genetic modifications to it, in this case put thylacine DNA inside of it, you can add these to an embryo or to an early embryo or egg, and those will be incorporated into a living adult animal. So this is kind of the route we're thinking about for using Tasmanian Tiger DNA. And I'll say that if you weren't familiar with this, John Gurdon and Yamanaka won the Nobel Prize for this in 2012. It continues to be one of the most widely used technologies for studying early embryos. Okay, so they've been working on this in Australia for a while, making these Dunner iPSCs. They had made them before I showed up. They found out that I was pretty good at analyzing certain types of bioinformatic data, and they said, we have piles of data. Can you help us determine if these are actually iPSCs? And so all the work moving forward is going to be my bioinformatic analysis of iPSCs they've generated. But I just did want to highlight in the last couple of months that I was there that Pranav and I were able to actually isolate Dunner embryonic fibroblasts and create our own iPS colonies. So this is a technology that I actually went there to learn. That's part of what a sabbatical is. And now that I'm back here, we're going to be doing this in the laboratory as well. So I'm really excited to be able to bring that back. But everything moving forward was not the cells that the two of us created in the last couple of months. They're things that have already been established. So while you can see these things are in a dish, these nice colonies, they look like iPS cells, these look like fibroblasts, you still need to figure out if they actually represent these cell types and if they have pluripotency, so they can develop into these things. The technique that's probably easiest to do for this is to profile the global transcriptome of these things. And so in this case, we extracted messenger RNAs from all the different iPSCs that were generated in the laboratory. These are shipped off to UConn. This is actually kind of coincidental. I went to grad school there, so I know Rachel really well. But Andrew and Rachel at University of Connecticut have a long-standing relationship where they're the sequencing core now. So it's kind of nice. I get to go back to UConn and know all those people. But anyways, samples are shipped off. They perform high-throughput sequencing, and in the end, what we have is RNA-seq data. And RNA-seq is basically what genes are turned on and off in the genome. We look at all the genes in one shot. And the questions we're going to ask are Dunner iPSCs reprogrammed? If so, do they express these pluripotency genes? And do they resemble known iPSCs from other species? And so at this point is when I acquired the data and started working with it. This is the first one of the first plots I generated for them. This represents some of the cell lines that they've developed, but not all of them. And this particular plot is looking at the variation in different cell lines. So if you start on the left-hand side up here, you'll see things as iPSC sendai. These are iPSCs made with one particular technology. This group of cell lines is made with a different type of iPSC technology. These are the fibroblasts from which these lines were derived from. This is organized by 4 principal components, so PC1, 2, 3, and 4. And what this is basically telling you is how similar or dissimilar are these samples from each other? What's the variation in there? A good example of why this experiment works fairly well, you can see that all the lines that are biological replicates look similar to each other here. All the fibroblasts and the similar lines look similar to each other here. So our reproducibility is good. And when you're looking at what actually drives the variation in these samples, for the primary driver of variation, you can see that whether or not a sample is induced pluripotent stem cell or sendai induced pluripotent stem cell or fibroblast is the primary driver of PC1. PC2, the sendai and the fibroblasts are somewhat similar, but they're very distinctly different from this class of iPSCs. And then we have a slight amount of variation here, but these are very minor contributors. The take home from this analysis is that while sendai iPSCs and fibroblasts are quite different, this other iPSC line, they look the same in the dish, but they're actually quite different when you look at the transcriptome. Okay, so that's really important. If you're going to make a pluripotent cell line that you're going to use in these reprogramming experiments, it has to be done correctly. So we already know right off the bat that these are quite different. When you actually want to look at what specifically is different, we want to compare the actual genes that are changing. In this particular case, this is a V plot. And so what we're looking at from left to right is how different they are when comparing sort of one fibroblast cell line to another fibroblast cell line, and whether or not the replicates are matched or significant, which is sort of above this line here. So if they're significantly changing, they're over in this quadrant or this quadrant. If they're changing, but it's not reproducible or not very significant, it's down in this quadrant. And if they're here, they're not changing at all. What you can see as a nice control is the fibroblasts are very similar to the fibroblasts. So fibroblasts are fibroblasts. No big surprise. If you look at the genes that are changing in these iPSCs compared to the fibroblasts, there are a lot of genes that are significantly upregulated in these Senda iPSCs. It's almost impossible to see in here, but they're actually labeled. These are known pluripotency genes that are upregulated significantly, which is great. In the fibroblasts, these are genes which are known collagen genes, which are known to upregulate in fibroblasts. So we can identify these lists of genes that are all changing significantly between these different lines. That's only marginally helpful. We have hundreds of these kinds of plots. We can identify the differences, but what's the actual meaning? So I thought it might be a good idea to ask, do our iPSCs look like iPSCs that other groups have generated? And so from Whitworth et al. 2019, they did a comparison between iPSCs that made in mouse, pig, platypus, and then they haven't made it for chickens yet, but these are just the embryonic stem cells. And they identified 39 key pluripotency genes that were upregulated in iPSCs. So I analyzed the gene expression of these 39 genes in this heat map here, comparing all of our samples. In this heat map, each row is a gene, each column is a sample. They were organized by hierarchical clustering, so that means they organize themselves into similarity. So all the Sendai iPSCs are more similar to each other. That's these 3 right here. All the fibroblasts are similar to each other. That's these 6 here, so on and so forth. I included 2 extra genes for analysis. These are known to be upregulated in fibroblasts and downregulated in iPSCs. So these are supposed to be like this. And if the color is red, it means it's turned on. If the color is blue, it basically means it's turned off. So for our known sort of like positive, negative control, depending on how you want to think about it, the genes that are known to be expressed in fibroblasts are turned on. In Sendai, if they're blue, they're turned off. Interestingly, for these other ones that I showed you on the previous PCA, they're actually more similar to the fibroblasts. Heat maps don't tell you a whole lot of information in terms of, you can't perform statistics off of them, but visually, when you look at this, you step back and take a peek, it's kind of obvious that the Sendai iPSCs, there's more red in these 3 columns, right? And that's suggesting that these key 39 pluripotency genes are more heavily or strongly activated for the Sendai iPSCs, whereas these other iPSC lines resemble somewhat of an intermediate state between these 2 things. And so we generated, I literally have folded hundreds of these heat maps looking at different lines that they've created, and we've been able to settle on some key iPSCs that we believe are really pluripotent and move forward with. Just to kind of like hammer this home one last bit, for this experiment, if we sort of add up and average the gene expression of all of those columns, on the left are the Sendai iPSCs next to their respective fibroblasts, and then these other iPSCs that were slightly different next to their fibroblasts. And when you try to analyze these using 2A ANOVA, we can see that there are statistically significant differences in the upregulation of these pluripotency genes of all these Sendai iPSCs versus all the others. So this was effectively a home run, right? So we were extremely excited to finally have a validated iPSC line, at least by RNA sequencing, that we can move forward with. The second part of this is looking at the epigenetic states of this. So that was just the RNA sequencing. In terms of epigenetics, we're going to consider histone modifications. And what we're really thinking about here is not only do the transcripts change, but are they actually marked by differential histone modifications? And so this, again, is data from the Takahashi paper in 2006. And it's really indicating that for histone modifications, in this case, H3K9 dimethyl, which is a repressive mark, there's enrichment on mouse embryonic fibroblasts. So this pluripotency gene is turned off and it's marked with repressive chromatin, whereas embryonic stem cells and iPSCs have been reprogrammed, and so it does not contain this mark. And then conversely, if you look at the same promoter, but for H3K, acetyl, which is a bunch of an activating mark, this activating mark is present in the promoter of embryonic stem cells and iPSCs, but is not present in MEFs when this gene isn't activated. And so we wanted to also look at this as sort of another state of this. And so using some genome-wide chromatin modification data that was present in the lab, but not really fully flushed out. I was able to identify 3 locations around this gene, which is the PAL5F1 promoter. PAL5 is actually OCT4, so it's one of those pluripotency genes. We looked at these 3 locations in the genome. We found that 3 of them had signals of enrichment for various histone modifications, K4, K27, and here's the replicas down here. And we decided to design primers to these locations to test whether or not this was true in actually the genome-wide libraries that we were making. I should note that down here, for example, is the OCT4 gene, this being the promoter region, and we're lining up with this peak. This is sort of our negative control over here, downstream region, which is outside of the gene. And this is probably like an enhancer region up here, so upstream. And so Pranav and I went, and this is one of the things that I brought over there. I taught them how to do these ChIP-seq or cut and run libraries, this way of analyzing these genome-wide histone organization. And we settled on 5 key epigenetic targets. So K4 trimethyl, K27 acetyl, 27 trimethyl, K9 trimethyl, and CTCF. If you've seen any of my work before, I spent a lot of time working on this particular protein. All of these are either associated with active chromatin on top or repressive chromatin, and their presence at the promoters or enhancers and regions of the genome will drive gene expression. And so what we want to do is identify whether or not these marks are present in induced pluripotent stem cells that we've made, as well as a bunch of other cell types to try to profile the genome and understand if we've completely reprogrammed them. The data down here is sort of, I would call this preliminary. This is qPCR that Pranav performed after we made the libraries. And he's showing some modest enrichment at this PAL5F1 gene, where, for example, H3K4 trimethyl is enriched in the promoter region, where it's supposed to be. This is, again, preliminary data, so this is actually at University of Connecticut right now for sequencing. So there's not error bars on this. But you can also see, for example, that K27 acetyl, which is supposed to be an active mark, is enriched at this upstream potential enhancer, this promoter. And then finally, it is interesting that this K27 methyl is also enriched as upstream region. Needless to say, we're well on the path to completing that part. So for project 1 conclusions, really it's at this point, catch the football, spike it. We're really excited. We have iPSCs that look very good, they look very pluripotent. The next step is to actually do genetic modification. So we're going to start with a GFP. They're going to start with integrating a GFP reporter, inject those iPSCs into Dunarts, and see if they actually are stem cells. Can they become every single kind of tissue? So we've identified the key iPS line. Now we're going to see if it actually can do it. And then immediately after that, start putting Tasmanian Tiger DNA in. For part 2, we're about halfway to the end zone here. We've established this protocol in the past lab. We've established the methods for doing the data analysis. We have generated the libraries, and they're actually sitting at the University of Connecticut right now, waiting to be sequenced. In the future, once it's complete, it'll allow us to identify key regions of the genome that we can actually target from, especially moving from one species to the other. Now, I'll just step back for just 1 second here. If you're thinking about taking Tasmanian tiger DNA and sticking in the Dunart, what pieces of DNA do you want to use? You have 3 billion bases to choose from. Which pieces are the most important or essential? This is the kind of information that's really important in making those decisions, identifying where the genes are, but also what other regions are functionally important by sort of epigenetic marks is also one of the key drivers of allowing us to figure out what components or what pieces of DNA we can move. Okay, so this project is ongoing, it's continuing. I'm still doing the bioinformatics work with these guys, and I keep getting emails and file uploads daily. The second part of this that I will go through quick, I promise, is actually driven by the work that I wanted to do when I brought there, and this is related to spermatogenesis. So in terms of techniques, we could use reprogram iPSCs we could take those iPSCs and put them into a nucleated egg and generate the embryo. But another approach we could actually use is to create a genetically modified sperm and then have that fertilize an egg. It actually could be a lot simpler than doing this in a dish. You can't just genetically modify a sperm, however. You actually have to modify its precursor cell, which is called the spermatogonial stem cell. And so the next bit of work I'm going to talk about is all about defining these types of cells in the donor so they can be genetically modified to try this sort of approach. I hope this guy looks familiar to some of you. Hopefully you remember this was your commencement speaker at Rowan University last May for our department. That is Aaron Augustini. He was a research student in my laboratory here at Rowan University. He's now a PhD student over in the University of Melbourne, working on this project. So while I was over there on sabbatical, we worked together for about 6 months or so on these projects. It was really awesome. And I continue to co-supervise him while he's over there. Okay, so for this project, this is the project. I'm using two approaches. I'm using some microscopy and some single-cell RNA sequencing. And I'm trying to ask what happens in Dunart spermatogenesis. And this is sort of the overarching goal, is to characterize this for fat-tailed Dunarts. But for my laboratory, I really want to know how DNA is packaged in systems. And so sperm is one of the most interesting cell types in an animal because it's so different. Remember when I told you earlier on in the talk that all your cells have the same DNA? Sperm doesn't. It's like one of the only cells that doesn't in your genome have the same DNA. It has half that DNA. To go from a full diploid genome to a haploid or half DNA genome, you have to go through a tremendous amount of cellular differentiation. And so that's depicted here, where you go all the way from a spermatogonial stem cell down to a mature cell. This happens in what's called a seminiferous tubule. And during this process, you actually completely repackage the genome. So you go from a genome that has almost complete histones, and it would be like all these yellow circles here, and it replaces these with small basic proteins called proteins. And this replacement allows the sperm to be compact, swim fast, but it also makes them transcriptionally inactive. So I was curious if the same type of organization, chromatin organization, happens in the Dunart, and so I performed the following experiments. Oh, I should mention, sorry, before I get there, that this is just an example of what these look like inside the testes. This is called a seminiferous tubule. Adult males will produce sperm their entire life from these spermatogonial stem cells here. They differentiate and migrate this way towards the central lumen, where they become mature sperm out here. This image is showing that histones are stained pretty darkly around the periphery, so these are normal cells. DNA is packaged with histones. And as you transition through development, these cells, these mature sperm cells, lose their histones, so there's no brown staining on the outside, and they only contain these proteins, which are the small proteins. Okay, so I wanted to look at this in Dunarts. So over at the University of Melbourne, they had some high-resolution microscopy facilities that were phenomenal. And so I was able to do this. This is a mouse sperm on the left. This is a Dunart sperm on the right. They are to scale and comparable in size. So as you can see right off the bat, given the same genome sizes, Dunart sperm, even though it's the same size animal's mouse, has huge sperm heads. That's really interesting because it's the same amount of DNA, so it implies that the DNA is packaged differentially. And one hypothesis is that they don't use proteins, but they actually keep their DNA packaged in histones. So we were able to perform this super resolution microscopy. This is with an area scan microscope, and effectively it just takes really, really, really small slices of a sample in Z dimension and at really, really high magnification, and then we can compile those afterwards to create a three-dimensional image of these. And we can do things like calculate volume, look at shape, make some really cool movies in Amaris, which we have on campus, by the way. So, and then look at the shape and structure of these things. And so hopefully, as I was talking, you guys noticed that the actual shape was wildly different, as was the size. And we start to stain these things, we can see that, for example, when we're looking at the DNA content, which is DAPI, the overall size of the Dunart versus the mouse sperm, and I kept these in the same actual slide field of view for their size comparisons and signal comparisons. While DNA is present for both these things, in this particular treatment, the protein stains really, really well for the Dunart, not so well for the mouse sperm. And when we're looking at histones, really the fact that the tail is staining here as well. There's almost no histone staining for both of these things. It's largely just background. So the noise is really, really high. The difference in treatments between the top and the bottom, in this particular case, DTT is a chemical you have to add when you're working with mouse or human sperm in order to decompress the DNA. So as the DNA is around histones, it's actually fairly loose. When it goes around proteins, it becomes much, much tighter. So the tighter the DNA gets, the less accessibility these antibodies have when you're actually doing immunofluorescence. And so if you actually want to do good immunostaining for these things, you have to actually add a little bit of DTT. When we do this, we see actually good staining for both protein for the mouse and the donor sperm. So it's not that the protein's not here, it's just that the mouse sperm is actually so highly compact and compressed that it can't be stained well. And again, neither of these, even with decompaction, stain really well for the histones. Maybe the mouse a little bit, but if anything, they're staining the mouse, but not the donor. So it's not like the donor is big because it's got a lot of histones. Okay. So the conclusion there is that the retention of histones are not the likely driver of the size differences in donor versus mouse sperm. One of the other questions you could ask is the actual compaction of DNA in mouse sperm caused by disulfide bonds, disulfide bond formation? So just a little bit of background for this. All proteins in your body are made up of amino acids. These amino acids vary in composition. In cases where you have two cysteines, these two cysteines can be bound together by a disulfide bond, disulfide bridge, and this will stabilize the structure and create higher order structures of these things. It also will compact a unfolded long protein in sort of a more compressed state. It turns out, interestingly enough, that if you compare all the different species, in this case, these are all just different animals, and you ask in their protein genes, are there disulfide bonds? You can't really do that too well without doing some biochemical assays, but what we can ask, are there cysteine present? For all these animals, including monkeys, Homo sapiens, mouse, pigs, the sheep, they all contain cysteines, and so all their proteins form these disulfide bonds, highly compact. If you look at all the marsupials down here, so this is the donor over here, these are various similar organs to the donor. They all actually lack these cysteines, except for the small group here, indicating that they actually cannot compress their DNA structure by forming disulfide bonds. So as an overview, if you kind of I generated this phylogenetic tree looking at this in a little more detail, and showed pretty clearly that all these eutherians form these disulfide bonds and have this high level compaction, whereas these marsupials do not have this case. So they're a unique case in which they do not have this higher order structure. Okay, so I think I just have a couple more slides about this compaction to kind of send this home. Remember I said that without DTT treatment, the mouse sperm is pretty highly compact, so it's smaller than the donor here. If we add DTT over time, it gets a little fuzzier, it's a little hard to see, but it actually is a little bit bigger here than this donor sperm. The donor doesn't actually change relative to the mouse in this case. Over enough time with DTT, this upside down drunken looking penguin here is actually a mouse sperm head that's been exposed to DTT for a while, where the DNA is actually decompressed because DTT affects disulfide bond formation. So it'll disrupt that. And the DNA has been allowed to spread out in this case, whereas the morphology of the donor head remains roughly the same. So this is really supporting evidence, again, that I was showing that the level of compaction in mice is largely due to disulfide bonds and compaction proteins, whereas in donors, this increased head size is probably a result of the fact that they don't have this formation. If we use a different dye for this, so not just DAPI, you can see roughly the same thing. So we can actually three-dimensional image these. We can use a MARS to create a three-dimensional volume around these. And so this is the shape of the DNA inside the sperm head for both the mouse and the donor. And then we can actually use MRS as well to calculate the internal volume the DNA occupies in these cases. And what we can pretty clearly see is that the donor does not change very much when you add DTT, when you reverse crosslinks, reverse the disulfide bonds. Whereas in mice, we have a significant increase in the size, and this is not the super addition of DTT because that wasn't quantifiable, but even when you add small amounts of DTT, you actually decompress the DNA and increase the size of it. Okay. So I want to make sure we got through that fairly quickly, but the key takeaways from that second part of the project was the work that I brought to Australia was looking at the chromatin condensation state in mouse and donor sperm. And what we found was that the presence of histones, histones were not present in donor sperm as they are not present in mice, and so are not the thing that contributes to the size differences. But in actuality, it's the absence of disulfide bonds in mature sperm that actually leads to the fact that the donor sperm are so big. Okay. And you know what, for time, I'm just going to do a couple little bits on this, and then I'll wrap it up. We also wanted to look at how these histones to protamines transition during mouse spermatogenesis. And so by comparing these two things, we could identify what key markers and genes drive the transition from these early spermatogonial stem cells all the way to these mature spermatozoa. During this process, this epigenetic transition, we go from DNA that's wrapped in histones and nucleosomes to DNA that's wrapped in protamines. This is kind of what I discussed previously. This is the case what happens in mice. We wanted to ask if this is also true in donor. So when does this transition happen? How does it happen? We did a whole bunch of testes slices. We profiled these cells in a variety of different tissues. And then we quantified what amount of histones, protamines, and other epigenetic marks were present at all these different stages. And this slide is just to show you that we can go from spermatogonial stem cells through differentiation, the left to right here, all the way up to mature sperm. And the next couple slides I'm going to show you, all these cells are present in one view in a testes slice. So you'll see cells that are like spermatogonial stem cells all the way to mature sperm. And all these sort of vertical columns, we can look at different stages by looking at different slices of testes. And so looking at this, whether or not histones or protamines are present in different stages, I have 4 stains for you here. I have DAPI, so this is looking at DNA. This is a seminiferous, it's a seminiferous tubule here, where these are spermatogonial stem cells here. This is sperm in the middle. This is an acrosome marker, so it's actually also marking the protein region in sperm. So this is actually a mature sperm. These are sort of pre-sperm here. This is protamines, so these are just mature sperm, which is why you see red over here. And these are histones. And so histones are mostly marked on somatic cells. You actually do see a little bit right here. These are the merged images of all these top bits. And what I hope you can even see on the projector here today is the fact that the red and the blue are actually overlapped with each other. And I'll show you this on the next slide and explain why that's important. So this is adding and subtracting these channels from a single image. And so here's DNA, here's histones. These are mature sperm, acrosomes over here. These are just the acrosomes. So this red is the protein signal, the blue is the histones. I'll leave it here for a second. So we can see clear localization of both the red and the blue channels, so both histones and proteins. And I'll say that this is completely unique to marsupials. So this has never been seen in any eutherian mammal before. At this stage, late stage in spermatogenesis, the transition from histone to proteins is complete, and there are no longer any histones. So this represents a really cool finding that we made over there. We're hoping to follow up on this a little bit. And it really suggests that, as opposed to the mouse model, we get a reappearance of these histones at later stages. So marsupial sperm are actually completely unique from this, from the mouse model. Okay, and I just want to conclude that by saying that, again, we saw the reappearance of histone proteins in elongate spermatids. and that this transition from histone to proteins occurs actually at a much later stage and can actually always persist all the way up to the epidermis, which is a post-testes tissue. Okay. And last but not least, future directions. So we've taken all of these testes samples from donors that we're looking at, and we've generated single-cell RNA sequencing libraries. They're currently at UConn being sequenced. What we'll be looking at when we get those data sets back, which will hopefully be in the next month or so, is how these proteins, like these histone proteins, or protein proteins here, are activated or repressed in different cells. In normal RNA sequencing, we can get just a bulk picture of what's going on, but in single cell, we can actually tease out the transcription profile of individual cells at all these stages and understand at what point actually do histones disappear and proteins come into play. And why this is important is not just from our research. This is also important because when you're trying to generate spermatogonadal stem cells and actually initiate programming in to become elongating sperm so that you can actually do that fertilization with the Tasmanian tiger, understanding how this transition occurs and what key markers or genes are activated or repressed in this process is how we'll actually be able to induce spermatogenesis in donors. Okay, so I just want to thank, I've had a lot of students over the years, but I want to specifically thank Aaron, who you'll know in both these lists here, both the Carone laboratory at Rowan as well as the past laboratory over in Melbourne. My 3 students who really held it down while I was gone, Yesenia, Lauren, and Juliana. Andrew passed, all of his incredible team over there that I worked with, but this is obviously not that entire list either. The O'Neill Lab for sequencing, and thank you for your attention today. Be happy to take any questions.
Students with some questions?
Yeah.
So you had touched on at one point about how when you de-extinct a species, it doesn't really bring back the genetic diversity of the species that had once existed. Do you think it will be possible at one point to simulate the genetic diversity by randomizing certain parameters of a genome to bring back some of that diversity and the resilience of the species?
Yeah, I think part of the idea about generating the IPS lines is not just to have one good one, but have multiple good ones from multiple different animals. So hopefully the genetic diversity also inherent in the donor genome will help provide some diversity to whatever organism that's been derived as well. Yeah.
Thank you.
Yep.
Are you also going to be able to replicate the environments that they've been in at one point? Like for the tiger, are you going to place them in a similar environment before that's more protected, or are you sort of...
So that's a really good question. That's a really good question because part of the reason these animals were chosen in the first place is because those environments still exist. It would not be ideal to bring a velociraptor back because that environment doesn't exist and we would all be food. But for the Tasmanian tiger, their environment did exist, sans the fact that we hunted them onto existence. There were some invasive species that also helped contribute, but it was largely human driven. So as long as we don't hunt these species, they still have the environments available for them to come back. Yeah, you see?
So if you're using the egg from another animal, is it from the dernet, right?
Yep.
Wouldn't it be more of a hybrid more than the actual tiger?
So great question. In this particular case, if you classic reproductive biology, you actually take all the DNA out of that egg. And so you're putting only brand new DNA into it. So you only get it from the somatic cell. But you are actually correct because in this case, because we don't have that intact thylacine nucleus, we actually have to make kind of a hybrid animal between the donor and the thylacinate. And the goal is really once we do sort of the epigenetic or genomic profiling, we can say, hey, what genes are present here that make the stripes or that make the tail or make the sides? And it should only be a handful of genomic changes that need to be made and not replacing the entire genome to create those phenotypic changes. But it will definitely be a hybrid, yeah.
I have one really quickly. Did you do any teratoma formation assays with your iPS cells or not yet?
They're doing those.
Okay.
Yep. So I came in at the point where they were like, hey, we have these cells in a dish. We're not really sure if they're iPS cells. Can you tell us which ones we should use for teratomas? So the question relates to if you take these iPS cells and you inject them into an animal, they should form teratomas. Those teratomas should form tissue-specific morphology, so all the different lineages, and you should be able to see, do these cells differentiate into all the like localized different types of tissues, and that tells you if they're pluripotent.
Kind of build off the hybrid thing. Are you intending on whenever you make this initial population of thylacines to have them interbreeding with each other, or are they kind of hybrids that you don't do that?
I mean, hopefully, right. So typically, if you're studying classical genetics, we think of hybrids as being sterile. So like horses and donkeys make mules, and then mules are sterile. In this particular case, because that's typically because of chromosome differences, chromosome numbers and size differences. In this particular case, the Dunart and the thylacine have the same number of chromosomes. So hopefully the hybrids will not have the sterility problem, as some of the other organisms do. You can follow up yet?
Yeah, and kind of just splitting hairs here, but with the fact that they are hybrids with other animal genetic material, would they not be considered the same exact species? Like when you had
That's correct. Yeah, so they would not be thylacine in the classical sense. You're not this de-extinction of the Tasmanian tiger, but in reality, it's creating a new organism that contains the Tasmanian tiger traits to be introduced, and hopefully the environment that still exists. Yeah.
So how are you going to guarantee the species survival away from like human interaction? Because you don't want to actually like domesticate like this type of animal. You want to be able to survive on the land, right?
Yeah, I mean, there's no way to guarantee that human beings won't do the right thing. So I think the promise of this technology, there's a lot of questions ethically about, should it be done or how can it be achieved? But the actual promise of these technologies is more lies in the process of understanding cellular reprogramming and how you could do these things. Those types of technological developments will also benefit sort of human health as well, that kind of thing.
Could you look at the structure of the histone tags, or like the structure of the chromatin before and after? So disappearance and reappearance during spermatogenesis.
You mean, did I look at the specific histone modifications that were present? Is that?
I guess so. So like, is it reprogramming? Is that why it's disappearing and then reappearing, or does it bring back the same epigenetic tag?
Oh, that's a really great question. So I'm glad you caught that. I was looking at the clock and thinking I don't want to keep people here forever. So I kind of went through this very quickly, but typically this is facilitated by histones being present here. These transition proteins kind of help facilitate the removal of these histones and the incorporation of protamines. In a mouse model, you completely lose the histones. The fact that the histones come back is incredibly strange, and whether or not these transition proteins exist in Dunart is unknown. So I'm going to look into, and I hypothesize that the transition proteins do not exist, and that these histone proteins have to come back and are required for this transition to occur, but they're actually a different type of histone. So a sperm-specific histone. So that's what I'm hoping.
Online, Jerry Huff asked, is this technique being done in birds as well? Do you know?
The dodo bird.
He said,
the trying.
He said there's definitely there's been a number of Hawaiian honey creepers that have gone recently extinct. And
so I will say that the other aim of this group, a lot of the funding is for the de-extinction project, but there's actually a lot of funding in this group. The large part of the team is for a conservation project. And so the IPSC work is to learn how to make these IPSCs for de-extinction, but actually what the past group is doing is collecting cells from a lot of different endangered groups and archiving and collecting those cells, making IPSCs and archiving. So if we have to do this kind of thing in the future, we'll have archived stem cells, kind of like the bio seed bank they have for plants, but instead for all animals. So that's a large part of the group is doing that as well now.
What are the goals? I mean, you said, we're not reintroducing thylacines. So who is setting the goals, and what are those goals of the things that are trying to be recovered? Because on some level, I would think some of these traits are going to be easier to break. It's like, but do I just end up with a donut with stripes? No, is that enough? Or
that'll be the first step.
Yeah, sure. But I mean, it's like, what is
That's a great question. So for the Tasmanian tiger, the Australian population is really excited because there's such it's like the Americans for the bald eagle. They like the Tasmanian tiger. For the woolly mammoth, it's actually a little more practical. Not the practical I should drive this, but it's more practical. There's almost no interest in creating a woolly mammoth per se. They actually want to create a hairy Asian elephant. Because in that case, there's nothing that really currently occupies that ecosystem in the tundra. So it's a completely vacant space because the organisms that used to occupy that have all died out. So there's open space for that animal to exist there. And so it's more about creating an animal that could exist in an environment that's devoid of an animal because we've removed it for it. And and I don't know much beyond this, but I will say that it's been posited that the woolly mammoth really helped by walking around the tundra to break through the ice and allow plants to grow through, and it was a good thing from that perspective. But it's probably set by Ben Lamb, who started the company, and then talked about by their board of directors. Yeah.
Online, Jesus Calixto says, "Hey Ben, great to see you in fantastic health and shape." And then asked two questions. The second was, do these fluorescent tags interfere with the structure or activity of the histones at all? So you're tagging them. Does that change their in vivo
I'm
function?
actually not tagging the histones.
Okay.
I'm just using, it's probably my fault because I just went through that so quickly, but we're actually using immunofluorescence to look at the native histones in these cases.
Awesome. And really quickly, his first question was, you mentioned injecting iPSCs into embryos as they may develop to seemingly reprogramming the epigenetic makeup due to their nature, pluripotent loss, tumorigenicity, etc. Can detrimental mutations arise in the pups, and does genetically and epigenetically uniform iPSC population address this?
Can genetic modification? I don't think it'd be very likely to have a large number of genetic modifications present in iPSCs. I mean, we sequence them all before we use them. And in terms of the actual animals themselves, I mean, nothing beyond, we wouldn't use a sick animal. So yeah, probably not a major concern for that.
Well, you probably mentioned that, but I just want to ask you, why did you choose actually the donor genome and look at the difference between the Tasman genome and the donor genome?
Right. So the Dunnar, even though it looks very morphologically different, small mouse versus big animal, their genomes are actually some of the most similar in marsupials. In terms of like, if you look at like a Dunnar, which is the size of a dog, and a dog, their genomes are actually very different. So as I said, right, a thylacine, the Tasmanian tiger, and a dog they look very similar, but their genomes are very different. So if you're thinking about taking small bits of DNA here and putting them into a dog or a cat, will they work? They probably won't work the same way in the same organism. Whereas in a marsupial, those bits of DNA are more likely to work in the same ways because those organisms are so closely related. There's other things like it was easy to work with in the lab because it's a small animal. And there's actually other things, and probably one of the most exciting things. So did Jurassic Park pop into anyone's mind when I gave this talk? Yes, of course. The other part of this is the matrix might also pop into your mind when I tell you that the most interesting part of the marsupial project is that marsupials develop mostly outside the uterus. And so a big part of Colossal's research is actually we can do these genetic engineering techniques because you don't have to have a giant thylacine growing inside of a small donor uterus for more than 13 days before it can be raised outside, only the size of a grain of rice. So you don't have to worry about large things like that. And that's why the marsupial model is so powerful in this particular case.
How close to the original thylacine do you want to be for it to be considered a success and be able to fill a spot in that ecosystem?
Yeah, that'd be great. Honestly, if we could put one gene in Dunarts and it had one phenotypic consequence, that was one gene from thylacine into Dunarts that had one phenotypic consequence, that would be a Nobel Prize achievement just to begin with. So these are very lofty goals in terms of genome editing. Once we get that would be super, super exciting. Yeah, it's a great question though. It's a hard thing. It's not. Yeah.
I actually have another question, but so I know that you were talking about the tails that you can find in the stones, right? So I'm not pretty sure about it. I'm not an expert with these things, but I just want to have a question. What are the functions of these cells? Like,
Right, so your whole genome, your DNA is wrapped around these histones all the time. And if the histones were all the same, it wouldn't actually have any functional consequence. But the histones are actually, those tails are chemically modified, and it's kind of like a little flag. And one can have a little flag that says, hey, I'm methylated. Why don't you come over here, polymerase or RNA polymerase, and turn on this gene? Another one might have a flag that says, hey, don't come over here, keep this gene off. So the differential marking of these histone tails allows you to program activation or repression of genes. For example, the OCT4 gene that I was showing you probably has activating histone marks at its promoter that says, turn this gene on. And non-pluripotency genes would be turned off in those cells, for example.
You briefly mentioned it, but what do you guys think with the
It's pretty much wild speculation. I'm not entirely sure, but it's very strange. It's only ever been seen. It hasn't been looked at other marsupials. This is the first marsupial that we looked at. And so I guess one of the things you want to do is look at different marsupials models as well. But then to follow up on it, we won't actually have a really good understanding of which histones these actually are until we get the single cell RNA sequencing back. And then we can say, oh, that histone's back in place at this time. Maybe it has some specific structure or function. So hopefully in about a month or two, I could have an answer to your question. And then there's just to kind of like follow it up. My laboratory has shifted mostly from doing a lot of experimental biology to a lot of bioinformatics types of things. So we're forming a long-term relationship with the PASC lab, and over the next year and hopefully much longer, most of the projects in my laboratory are going to be, hey, let's work on the data you guys have generated. I really like the model so far of having another group pay for all the experiments and then getting all this beautiful data back and driving the conversation and then also helping interpret the results. So if you're wondering what I'm doing, I'm spending a lot more time over in the computer side of things than the experimental wet lab, but that's sort of the fun part about sabbatical is changing it up a little bit.
Very cool.
Cool. Thank you guys.
Let's all thank Dr. Carone one more time.